Well, thank you, Brenda. Welcome, everyone. I'd like to thank Jean Christie as well and Lee Daniels for helping to set up this session. Today I'm going to be talking to you about redoxamorphic reactions and describing redox features. Redox is an abbreviated name that we use instead of repeating the longer redoxamorphic term each time. Again, I'm Dr. Galbraith from Virginia Tech, and I'm an Associate Professor of Soil and Wetland Sciences. Welcome, everybody. Well, we have three learning objectives for today's webinar. First, we'd like to explain the definition of hydric soils. We'd like to be able to recall the basic redox reactions, and we'd like for you to be able to inspect and describe redox features following this webinar presentation. Well, let's start with the definition of hydric soil. The hydric soil is a soil that, part one, formed under conditions of saturation, flooding, or ponding. Part two, the saturation, flooding, or ponding took place long enough during the growing season to develop, part three, anaerobic conditions, and part four, in the upper part of the soil. And so we'll discuss these in the four components. So when we say that a soil formed under the conditions of saturation, flooding, or ponding, that means that they may no longer be under those conditions. As long as they formed under those conditions, areas that have been artificially drained can still be considered hydric soil, provided that they meet all of the rest of the criteria involved. And if they contain the morphological evidence that we will be reviewing today and in subsequent webinars, then we can assume that they did form under those conditions, because soils that did not form under those conditions of saturation, flooding, or ponding do not contain the morphology that hydric soils contain. Now let's discuss part two, which is long enough during the growing season. Now this is a moving length. It is set in definitions and regulations, and depending on whether you are talking about different regulations concerning either restoration of a wetland or creation of a wetland or just identifying and delineating wetlands on the ground, there is a different length of time. In general, we're talking about 21 days consecutive anaerobic conditions, or if we're talking about flooding or ponding, then we're talking about 7 consecutive days of flooding or ponding, and that needs to be something that takes place frequently, and so not just occasionally but frequently flooding, and I think that means once every other year on average. Earlier I said that we needed 21 days of anaerobic conditions. We actually need 21 days of continuous saturation, which means that the water pressure is 0. But let's talk about part three, which is anaerobic conditions. And in this case, we're talking about the absence of gaseous oxygen. And this takes part due to microbial activity, and we'll discuss this in more detail later in the webinar. And then when we talk about the upper part of the soil, we're generally talking about the upper 30 centimeters of the soil, and there is a little bit of variability in this when we get to soils of extremely different textures, such as those that are very, very sandy or very, very high in organic matter and carbon histosols and other soils like that. All right, well, let's talk about redoxamorphic features and redox. Redox is an acronym that's short for reduction and oxidation. And redoxamorphic features are the characteristics that form as a result of redox reactions and as a result of soil solution movement. So that's how we get redistribution of soluble byproducts, and that's why we end up with either depletions of iron, manganese, sulfur, and other minerals from certain zones in the soil, or where we may get concentrations of those, is through the movement in the soil solution. I will go through the basic redox reactions. Now, I don't have any chemical formulas in this webinar, and I don't have any charts or figures or graphs that show these reactions and the processes that go on. It's actually quite complex, and there are a lot of variables involved. And so I tried to simplify it by converting it all to a text explanation. But I would encourage you, if you'd like further explanation, to consult a textbook such as the new version, the second edition of Wetland Soils, or the Basic Soils Handbook, or any other soil guide. All right, so redox reactions involve the transfer of electrons between two atoms. And these transfer occurs simultaneously. And so as one atom donates electrons or gives them up, then another atom simultaneously gains those electrons. The term we use for the gain of electrons is reduction. And when we get reduction is when the charge gets more negative because it received a negative electron. Oxidation is the opposite. Oxidation occurs as atoms lose or donate electrons, and in this case the charge gets more positive. Reduction is mostly microbial driven. Now, this may not be completely true in systems with sulfides or sulfates, but in most other systems we are talking about microbial driven processes. And essentially the electrons are generated during the metabolism of sugar and carbohydrates by microbes. And this is why part of the hydric soil definition and other parts of the hydric soil explanation that you'll hear relate back to growing season, is because of the dependence on microbial processes to produce these redox reactions and then the subsequent morphology that forms as a result of the redox reactions. All right, as far as reduction goes, elements have varying affinity for the electrons. And so those that have the greatest affinity for the electrons that are released during decomposition of organic matter are said to have high redox potential. And oxygen there's a sequence of atoms that we discuss because they commonly produce morphology or characteristics that we can measure in the field. And those are oxygen, nitrogen, manganese, iron, sulfur, and carbon. And of those elements, the one that has the highest redox potential is oxygen. Now, therefore, gaseous oxygen is reduced first in the soil. And so long as the gaseous oxygen is not replenished, the soil solution becomes anaerobic. So the first step in the sequence is saturation. The second is the reduction of gaseous oxygen. Following that, we get reduction in sequence of nitrogen and manganese, and then iron, and then sulfur, and then carbon. And it takes place in a reverse order during oxidation processes. Manganese and iron lose color and become soluble when they are reduced. So they go from a crystalline form into a soluble elemental form. And then now the soluble iron and manganese move with the soil solution. Now, if most of the manganese and iron leave a particular zone in the soil, it becomes depleted of those elements, and the zone is said to be a redox depletion. On the other hand, nitrogen, sulfur, and carbon produce gas when they are reduced. Rather than becoming soluble, they turn into a gas and do not become part of the soil solution. Now, oxidation of the reduced element does not require microbes. It can happen in the presence of microbes, but it does not require it. It could be a simple chemical oxidation that occurs upon exposure to gaseous oxygen, and it can also take place as part of the weathering processes that are naturally occurring as bedrock and other minerals weather in the soil. Now, as gas, oxygen, or nitrates or sulfates are introduced into the soil solution, the redox potential rises. A soil is said to be aerobic when oxygen is able to remain in the gaseous form. And when the redox potential is high, then oxygen is able to remain in the gaseous form. Elements reoxidize in reverse order to their reduction sequence, as mentioned earlier. And as they reoxidize, manganese and iron precipitate as crystalline oxides and oxyhydroxides. And each one of these has characteristic colors. And this is what allows us to use oxidized manganese and iron precipitates as redox features that help us identify the soil as a hydric soil. Now, some of the gaseous forms can be used as redox features, in particular hydrogen sulfide gas, which produces an unpleasant rotten egg odor. That can be used as a redox feature as well, although it's a gas and is invisible to us. But as long as our noses are working out there, then we can detect the presence of reduced sulfur. As the soil solution moves and the soluble iron and manganese move with it, they become relocated as long as the soil solution moves. In other words, if the water is moving in the soil and is not stagnant. Now, the relocated accumulations of iron and manganese are known as concentrations or redox concentrations. If the process continues and iron and manganese accumulate in the same place for a very long period of time and the concentration gets high enough, they can actually become cemented. And we'll discuss that a little bit later. Well, let's take a look at some of these redox features. The figure on the left shows a mass of soil that is largely oxidized and has a characteristic light brown, beige, or orange color to it. But you can see zones in the soil that are very light silvery gray. And in fact, the zone going through the center of this soil mass has some carbon in it, a dead plant part. And next to that dead plant part, which serves as a food source for microbes, we have reduction and depletion of the iron and manganese. What I cannot tell by looking at this gray zone around the root is whether or not there is still iron present and it is in the reduced form, or whether the iron has actually completely migrated out of the zone. Now, the picture on the right shows redox concentrations in the center between the aggregate surfaces. Now, here we can see the dark lines, the little black lines that go through the photograph, and you can also see what looks like little pinholes in the soil. Those are called pores. These are called macros together, both the pinholes and the edges of the aggregates. This is typically where the water travels first until it soaks into the centers of the aggregates. This particular soil, we can tell that water stays present in those cracks and in those pores for long periods of time, long enough to produce reduction of the iron and the manganese. Well, let's talk about the features. Let's talk first by talking about the zones or surfaces of reduction. where we can get the depletion of manganese and iron. Now, when we talk about zones or surfaces, zones would be three-dimensional, whereas the surfaces would be two-dimensional. And this surfaces we're talking about would be the linings around the pores, the linings around root channels, and the linings between the surfaces where two aggregates come together in the soil. We can also get zones of reduction. Inside those zones of reduction, if we get repeated removal of iron, we can actually get depletion of clay through disruption of the clay particles. In other words, we can get such extreme repeated reduction that we can break the clay particle and it can decompose chemically, and then it can be moved out, the components can be moved out, and you can lose clay content. Now, we can get concentration of manganese and iron oxides and oxyhydroxides either on those same surfaces that we just talked about, inside pores or channels. We can get them as masses inside of PED interiors, and PED is another name for a soil aggregate. And we can actually get cemented forms as described earlier if the concentration gets high enough. Well, let's look at the depletion zones, and these are going to be the zones that surround the macropores shown in the picture on the left. So the depletions that occur only on the surfaces are two-dimensional, but as those depletions increase in size and move into the soil mass around them, they become three-dimensional in size, and I call those zones. On the right-hand picture is a macropore, a vertical macropore in a soil that is probably dense and has very slow hydraulic conductivity. And we often find preferential water flow in these vertical macropores, and you can see the extreme depletion within the center of that macropore. You can also see that iron has migrated away from the center of the macropore and toward the mass of soil on either side. What I could say is that if you get continued reduction and removal of iron in these vertical macropores, this is where you would likely find a loss of clay as well. Well, here's a depleted surface. So if we pulled the soil apart and opened it up where it naturally separated into aggregates, and we looked on the surface of one of those sides that was exposed, if we see this bright silvery gray color, then we know that it has been depleted of oxidized iron. You can also see traces along where the pores and where the roots existed along this surface. Here's a depletion inside of a pore, and in this case the pore is actually a channel made by an earthworm. And so the earthworm, as it migrated down the soil, it has a mucus that is released to help it move through the soil. And this mucus is a very easily digested form of organic carbon for the microbes, and so they can easily metabolize the organic matter and then donate electrons and reduce the iron right next to the pore. Here's a zone in sandy soil, and we can see the gray silver depletion zone in the center of this mass of soil, and you can also see the areas where the iron has migrated to and reprecipitated as a new iron oxide compound, the bright red zones. Here's a look at some of the clay depletions, and in this case these are the surface between prismatic structure in what's called a fragipan. And so if we get soils that have a fragipan and also have anaerobic conditions above the fragipan, then we will get reduction in the pores going down between these prisms, and eventually we get clay loss as well. Now, let's describe what are probably the most common features seen in hydric soils, and those are soft masses. And these are concentrations of iron and manganese. The ones that are the darkest, shown where the blue arrows are the ones that have the highest manganese concentration. We almost never get pure manganese oxides in a soil. We almost always get some iron in association with the manganese, but we can get, on the other hand, we can get almost pure deposits of iron oxides in the soil. So just in general, when we see crystalline forms, in other words, this is not charcoal, this is not organic matter, these are crystalline forms in the soil that are very dark in color, we know that they have a high manganese concentration. The iron in the lower inset right, there are various colors of iron, and each of those is likely a different iron oxide or oxyhydroxide compound. But those are the ones with the nice orange and red color in the soil. Now here's a look at soft masses in sandy soils, and you can see that they're not as distinct. They're not as easily seen. I believe Lenore went over distinct contrast in last week's presentation, or the first week's presentation. But you can see, if you look closely, that there are some reddish colored zones in the sandy matrix in both the left and the right side. What you do not see is surfaces coated with oxidized iron and manganese in these sandy soils, because there are very few surfaces to begin with. There are very few macropores. There are very few PED surfaces because there's very little soil structure. Here we can see iron oxide that's precipitated around the edge of pores. So we know that oxygen has these pores on both pictures, and then the reduced iron that was present in the soil next to the pores became oxidized through exposure to the gaseous oxygen. The same can be found around certain root channels. If the root channel is open and oxygen enters the surface, or if carbon dioxide or oxygen is expressed by the plant through the root, then we can get oxidized, let's see, we don't use the term oxidized rhizospheres anymore, but we can get precipitated iron right next to the living root or next to an empty root channel. Here's a look at some redox concentrations of iron and manganese on surfaces. So this is where aggregate surfaces come together, and you can see that the zones that are circled are redder than the background color of the soil, and so they do have a concentration of iron. They often are associated also with clay that gets deposited in these oxidized conditions. Now here's our cemented forms. We have nodules and concretions, and there's not much difference between the two. Nodules have no concentric rings. If you break these open with a rock hammer or a sharp knife, if you notice concentric rings, they're called concretions. But that's a very minor difference between the two. The important thing to know is that these are highly cemented, and it's hard to know whether or not they are forming or degrading in today's climate. We know that they form through redox processes, but it takes careful investigation of these. You have to look very closely to the outer edges or rind around these cemented forms in order to decide whether or not and interpret whether or not they're actively forming in today's climate. All right, now as we describe these features, we need to tell several things about them. As you're taking field notes in the soil in the field, we need to know what type they are. First of all, are they depletions or are they concentrations? Tell us where you found them. Did you find them lining a pore, as a soft mass on PED surfaces, for example? Tell us about the color. Tell us about the size. Are they fine, medium, or coarse? And these terms are all defined in the field book for describing and sampling soils, version 3.0, that's available free from the USDA NRCS Soils website. We call it the purple book if you get a printed version because it's got a maroonish purple cast to the color. It's a waterproof pages. And the last thing we would like to describe is the contrast. So what is the contrast between redox features and the matrix of the soil? Are they faint? Are they distinct, or are they prominent? And again, that contrast has been described to you earlier in Lenore's lecture. And we will be saving questions for later, I believe, and so this ends my portion of the program, but I will stay online and be available to answer questions later on in the program.
Thank you so much, John. A wonderful presentation. I learned a tremendous amount myself. I'm going to hand over the presenter tools here to Bruce.